Assalamu alaikum. Welcome everyone. Today we will discuss mumps virus, which is an important human pathogen belong to the family called the Paramyxoviridae and the genus Orthorobellovirus. Although vaccination has greatly reduced the disease, still now mumps continues to cause an outbreak even in vaccinated population. As medical laboratory scientists understanding about the biology of the virus and correct laboratory diagnosis is essential for the patient management and outbreak control. During this lecture, we will cover virology, pathogenesis, clinical feature, laboratory diagnosis, prevention, and the role of the laboratory in the surveillance. Let's start by the learning objective. By the end of this lecture, you should be able to classify a mumps virus and describe its structure, explain how a virus replicated and why this is important for laboratory diagnosis, understand how the virus causes disease and spreads throughout the body. Also able to choose the correct clinical sample and know the best time for the sample collection. Also able to compare different laboratory techniques from molecular, serology, culture, especially for their advantage and limitation in vaccinated individuals especially. Finally, able to understanding the important role of medical laboratory scientists in diagnosis and outbreak investigation and the public health reporting for these infections. Let us start to give you introduction about this virus. Mumps is an active viral infection, mainly affect the salivary gland, especially parotid glands, causing painful swelling of this gland. Before the introduction of the MMR vaccine, it stands for the measles, mumps, rubella vaccine, and mumps was one of the most common childhood infections worldwide. Today, the number of the cases has decreased due to the use of this vaccine, but still an outbreak occurs, particularly, especially in university and crowded setting and military camps. An important point of this virus is that many infected individuals do not develop parotitis. Around 20 to 30% of the patient have no symptoms at all. The incubation period usually runs from 16 to 18 days, although it may range from 12 to 25 days because a vaccinated individual may still become infected. For that, we need a laboratory confirmation of this patient, or a laboratory confirmation of this patient is important than relying on the clinical symptoms for diagnosis of the vaccinated patient which is infected by the mumps. Here is a general taxonomy and classification of the virus. The virus belongs to the order Almononegative virus, which contain a virus non-segmented negative sense RNA genome. It is a part of the family Paramyxoviridae, which also includes this family also measles and human parainfluenza virus. Within this family, mumps belong to the subfamily of Ortho Paramyxoviridae and the genus Orthorobellovirus. The virus of mumps has only one serotype, which is good for vaccination because the antibody against one serotype generally protect against all the strains. However, there are 12 recognized genotype different, which are useful for the epidemiological study and the outbreak investigation, not for vaccine. Human are the only natural reservoir of the mumps. There is no animal reservoir which making a vaccination through vaccination theoretical is possible to control an infection. Here you can see structure of the viron. of the mumps viruses. Mumps virus is an evolved virus measuring about 150 to 300 nanometer in the size. Because it has a lipid or involved, that may be maybe this virus is sensitive to heat, the detergent, and the disinfectant. On the viral surface, we found two types of glycoprotein. The first is an NH protein, here which bind to salicylic acid receptor in the host cell and also help in the release of newly formed viruses. The second protein, glycoprotein, which found in the involved, is called the fusion protein or F protein, which allows virus to fuse with the host cell membrane and also causes the formation of essential cell or multinucleotide cell. Under the involved, there is a matrix protein. This matrix protein provides structural support and helps assemble new virus particles. Inside this component, we find the helical nucleocapsid containing negative sense RNA genome associated with the nucleoprotein and the viral polymerase protein. Remember that in the two protein NH protein هو responsible for the attachment, while F protein is responsible for the membrane fusion. This is a common example. This is a picture of the mumps virus under transmission electronic micrograph. You can see this is a helical genome. Here we talk about the genome organization. The mumps has a single strand negative sense RNA اللي هي قلنا the weight is 15.4 kilobase long. Since it is negative, because this virus it is negative sense RNA, the genome cannot be translated directly into protein. The virus first produce a positive sense messenger RNA using its own RNA dependent RNA polymerase. The genes of this virus usually arranged in a specific order. اللي هي start from 3 ladder اللي هي ال N gene and B and M F SH NH and L. These genes, all of these genes are transcribed one after another, one transcript and another another until the end of the 5 prime. Starting the transcript started from the C prime end of the genome. Another feature of this gene, important of this gene, is that in the B gene, you can see here it's the B gene undergoes by RNA editing, producing a 3 different protein. Here maybe LB, V, or an I. LV it is the most important one because it helps the virus to escape the body's interferon response. That means it is help the virus to escape from the immune response. Here in this table, you can see different structure and non-structure proteins of the virus. Here, this is summarized of a different protein which is produced by this genome. اللي هي start from N اللي هي nucleoprotein. النيوكليوبروتين اللي هي surrounded and protect the function. It is the need to surround and protect the viral RNA. And B protein help the viral polymerase during the RNA synthesis. والL protein اللي هي viral RNA dependent RNA polymerase are responsible for the post transcription and replication. and the M protein اللي هي inner involved leaf flag اللي هي M protein it is or a matrix protein اللي هي contain the nucleocapsid and protect it from outside to viral involve and help assembly of a new virus. F protein اللي هي help a virus fuse with the host cell and also to form a syncytium formation here multinucleotide cell, which is an important cytopathic effect you can see in the infected cell culture. An HM protein bind to the cyclic acid receptor on the hot cell and also help release a newly formed virus from the infected cell. والSH protein اللي هي found in the involve اللي هي مهمته الأساسية إنه modulation and it is commonly used for the viral genotyping. Finally, an V oral I protein اللي هي help virus to evade the host immune response by blocking the interferon signals. This is a general structure and non-structure protein which is translated by the genome of the virus of the mumps. Viral replication start from the attachment, fusion, entry, transcription, genome replication, and assembly, finally unbundling. Mumps virus complete it is entered. You can see here in the picture. Mumps virus complete it is enter replication in the replication cycle in the cytoplasm. That means this mumps virus replication cycle occur in cytoplasm, not in the nucleus. The first step of this cycles is started by the attachment of the host cell by a specific receptor. NH protein اللي هي glycoprotein found in the involved binding the cyclic acid receptor which found in the respiratory epithelial cell. After that, the F protein mediate the fusion between the viral involved and the cell membrane of the host cell, allowing nucleocapsid to enter the cytoplasm. And viral polymerase begin transcription, producing a viral messenger RNA. After enough producing viral protein produced, and virus switch from transcription to genome replication, producing a full length mineral antigenomes followed by the new negative sense of genome. After that, the viral protein and the genome assembly under the plasma protein under the plasma membrane with the help of the M protein. And after that, finally, in the final step, the mature virus leaves the cell fiber, acquiring their lipid involved from the host membrane. Mumps, unlike herpes virus, mumps virus never enters the nucleus and does not have DNA stage. In the cell culture, an infected cell usually fuse together to form a multi-nucleotide gain cell, which you know as a synethia. Synethia due to the action of the F protein, which is a help in the diagnosis of the tissue under microscope. Here is the synthesia formation caused by the mumps viruses in the cell culture. Here we talk about the epidemiological and pathogenesis of these mumps. Globally, before the MMR vaccine became available, mumps was one of the most common childhood disease. The pre-vaccine area and post-MMR introduction. After to introduce of MMR, incidence of the infection fell over 99% in the country with standard high dose coverage. يعني مع الدولة اللي عملت كفرنج لكل البوبوليشن حقنا 99% نلقى حصل فيل في الانفكشن او الديستريبيوشن للانفكشن. الريكرنس باتن للانفكشن او الاوتبريك occur in close contact setting. بنلقاها دايما في اليونيفرسيتي، في الكامبس، في الميليتري براكس اللي هي large attribute to waning. All this recurrent الابدemic due to waning vaccine induced immunity rather than vaccine failure. يعني community or population who vaccinated and after vaccinated there is a virus, there is a mumps outbreak occur in this community or population, not due to the fail of the vaccine. This is due to waning of the vaccine, the decrease of the immunity which winning by the immunity. Usually, mumps occur in the winter and the spring, big in the temperature climate. دايما الانفكشن بتاعت الممس تحصل وين؟ تحصل في الوينتر والسبرينج تايم. Here is a transmission Here is the transmission way. The mumps is transmitted primarily by the respiratory droplet or by direct contact or by vomits. Respiratory droplet like what? Like coughing or sneezing or talking, close contact, saliva or sharing of equipment, cups or cigarette, vomits. This is the virus found in the cervix for a long time. For sometimes with the respiratory situation. All vomits contaminated by respiratory situation, we found that virus in this area. Here pathogenesis, entry, primary, and replication. Infection begin when the respiratory droplet containing a virus is inhaled by the patient. First, a virus reaches the upper respiratory tract. After that, the NH protein attached to silical acid receptor on the epithelial cell. F protein help the virus to enter these cells by membrane fusion. After entering the cells, a virus begin replicated locally in the cytoplasm, not reach a nucleus. Replicate locally in the respiratory epithelial cell nearby cervical lymph nodes. After that, the virus enter a bloodstream, which causes a primary viremia. Through this time, the blood circulation spread virus to many organs before the symptoms appear. After that, the virus related to a pituitary gland and causes swelling of this gland after some time. Here is the pathogenesis, systematic spread, and tissue present. In the secondary viremia, after secondary viremia of the simulation of the virus's organ, virus reach the spherical receptor of NH attachment when in the specific cell. السبيسيفيك cell هي وين بتكون؟ في البورتيريال gland. هي most common affected organ, usually causing the typical painful swelling called the bursitis. The virus also can spread the central nervous system, causing aseptic meningitis and rarely encephalitis. Also can infect the male and reach the testis, may become infected and resulting in orchitis اللي هي infection. The female also may cause orchitis, which is less common in the female. After that, also can infect the pancreatitis, also can infect the inner ear. Maybe in the complication, one of the most important complication leading to hearing loss. BAM. Here is a clinical feature of the infection of the bursitis. The most recognized feature of the mumps is the bursitis, and here is swelling of the gland of the saliva or inflammation of bursitis glands. Before swelling develop, a patient usually experiences a prodromal phase lasting for 1 or 2 days before swelling appear. During this stage, patient may have low-grade fever, headache, muscle pain, loss of appetite. After that, one or both bursitis gland becomes swelling and painful after 2 days or 1 day. In the most systematic patient, بنلقى swelling دايما بيحصل لها eventually become bilateral, not one. Although it starts by the one side large. Patients frequently complain of the pain of swallowing and eating because this moving it stimulated the saliva production. The swelling usually reach a maximum. It is maximum size after about 3 days and gradually disappear within 1 week. يعني swelling ده بيكبر في 3 أيام وبعد after 1 week بيحصل له شو disappear. Usually clinically the swelling causes loss of the normal angle of the jaw. Here you can see this baby have a no angle of jaw ما في حرك بتاعه, which is a classical sign of the mumps. دي واحدة من clinical signs بتاعة the mumps infection. Here you can see this baby. We have a right gland infection. Complication of the mumps. The complication of the mumps is a two type, maybe reproductive or maybe neurological. طيب ممكن تعملها أوركوتايتس في الميل وممكن تعملها أوفراتايتس في الفيميل also may cause الاسبتيك meningitis or الانسفالايتس very rare والسنسor neural hearing loss اللي هي in the one ear. This when occur it may become permanent for the all life. This is one of the most complicated of the infection with mumps. The other complication, the other organ may be have a complication come from the infection by the mumps is pancreatitis, arthritis, and myocarditis nephritis. These are less, not common, and the pregnancy first trimester of the infection associated usually with the miscarriage. But there is no congenital malformation syndrome like a rubella فرشتك نلقى إنه الفيميل pregnant which is affected by mumps في الفيرست ترايمستر دايما بيحصل لها miscarriage لكن ما بيكون في خطورة على البيبي وبيحصل له شنو congenital malformation syndrome like a rubella. Here you can see human host immune response for clinical infection. Not everyone infected with the mumps develop typical swollen parotid gland. Not anyone, not all the person infected by the mumps develop typical swollen parotid gland. Usually about 20 to 30% of the infected people by the mumps are asymptomatic, while others have only mild respiratory symptoms. Even without symptoms, this individual can still spread the virus to other persons. This is the one reason why laboratory confirmation is important during an outbreak. Let's talk about the immune response against this virus. Virus defense is a innate immunity system which produces a type 1 interferon to limit viral replication. However, the virus produces a V1I protein which interfere with the interferon signal and help the virus to survive. After that, develop adaptive immunity, the IgM antibody appear about 3 to 5 days after the system onset. After that, the IgM when the IgM appears, that indicates that is a recent infection, but may be absent in vaccinated individual. The IgG antibody develop later and usually provide long-lasting immunity for a long time. After that, the T lymphocyte cell will develop to eliminate the infected cell and contribute to long-term immunity. All the mumps is the classical cause of parotid swelling. There are other diseases can produce the same swelling of the parotid gland, like a viral infection such as the influenza virus, the Epstein-Barr virus, or cytomegalo, or HIV, or Coxsackie viruses. Also, there are some bacteria can cause the parotids, and here like Staphylococcus aureus. There are other non-infectious other syndrome non-infectious اللي هي سيجيرنون syndrome والسكاردويدس syndrome والدرug reaction والكحول-related infection. Other lumps miming swelling اللي هي cervical hepatitis والdental abscess. All of this is all other parotids infection. That for swelling or inflammation, for that we need a laboratory to confirm is this mumps or the other pathogen infected or the other cause of the infection. Let's talk about the diagnosis of these viruses. Accurate diagnosis depends on the three factors. Depend on the correct sample, correct sample collected, and the time and the right time to collect it of the sample after the symptom onset. The third point is the choosing of the most suitable laboratory method for diagnosis of this patient. Today now, real-time PCR is one of the most suitable method for diagnosing acute mumps infection. However, serological testing and the viral culture still have important roles in a specific situation. Let's to see different samples which you collect. A proper sample to collect it from to diagnose a patient who infected by mumps هي buccal or patroid duct swab. Before collecting السواب gently بنعمل مساج للباثرويد gland about 30 seconds to increase the amount of the virus enter the duct. After that we introduce the virus. After that introduce the swab gently in the pathroid gland and moving out inside the duct to check. Urine also may be collected, can be collected because the virus may be shelved for a longer period than in saliva in the urine. CSF involved in the suspected individual who is suspected have infection in the central nervous system should be collected for diagnosed by the PCR. Also, blood sample we can collect it to test it an IgG or IgM antibody. A sample must be transported in a viral transported media and kept in refrigerator for at 2 to 8 centigrade and proceed quickly. For the long storage, we put it in -20 and avoid freeze soul cycle many times. Okay. Here in this slide is summarized the recommended diagnostic approach. First, identify a patient. First, we need to identify a patient with Virus or another compatible clinical syndrome by the clinical symptoms, especially if there is a history of exposure or ongoing outbreak. After that, we collect the buccal swab, urine sample, acute serum sample at the same visit. All these samples collected at the same time. We perform a PCR for a buccal swab because it's a suitable test during the acute infection. If a PCR is positive, the diagnosis of mumps is confirmed regardless of the vaccination history. At the same time, perform an IgG or IgM. With IgM negative, when an IgM negative, that does not exclude the mumps infection in vaccinated patient. Because their antibody response may be weak or absent because this patient give this virus before this time, which lead to develop to an IgG for a memory cell. Finally, even confirmatory cases should be reported. أي confirmatory cases should be reported to public health authorities, and the positive sample should be sent for molecular laboratory for genotyping during an outbreak investigation. That means we have steps for diagnosis of mumps patient. We start from the clinical suspicion and collect the right sample, 3 samples, swab, urine, 1 blood. After that, we do PCR, a serological test, and finally a report notify health authority for the result. Molecular detection. For the molecular PCR is considered a gold standard for the diagnosis of this acute mumps infection because it's fast, sensitive, and affected by the vaccination. PCR detects the viral RNA, making it more sensitive than the other testing. Unlike an antibody, a PCR do not affected by the previous vaccination. When we detect an antibody, this antibody is affected by the previous history of the vaccination. The most common target gene who في PCR is the SH gene, which is also used for genotyping. When N gene and the N gene, we use the SH gene detected by a PCR, and the SH when N gene used for genotyping. The timing of the collection sample is the most important. Highest sensitivity is during the first 3 days after virus begin. As the time passes, the amount of the virus decreases and the PCR sensitivity is declining. يكون PCR used in the acute case. Therefore, negative PCR result obtained late in the illness does not completely exclude an infection. Always integrate the PCR together with the patient system vaccination history and the sample collection. دايما بنعمل integration result مع كل الفاكتورز اللي احنا خدناها. History of patient symptoms and date of the collection of the sample. Early sample collection give a high PCR sensitive usually. Virus culture. Before the molecular method became available, viral culture was considered the reference method for the diagnosis of mumps. But today, the culture technique is used for the research, surveillance, and genotyping rather than a routine for them. The common cell line used for the culturing of the mumps virus is the virus cells, LLC-MK2 cells, and primary monkey kidney cells. And infected cells develop and form characteristic cytoplasmic effect, including a cell rounding and formation of multinucleotide gain cell. Another laboratory technique used for detecting this growth of the cytoplasmic effect is the hemoabsorption assay. Hemoabsorption assay, which is in this test we use big RBCs, which these RBCs attached to a viral NH protein. This allow infection to be detected even before previous or obvious cytoplasmic or appear of cytoplasmic effect in the tissue. A viral culture requires 3 to 10 days, while PCR requires few hours. For that, we're using PCR for diagnosis of the patient, and the virus culture for the research, surveillance, and genotyping. Here is the serological timing or serological test, IgG or IgM. Serology usually measure a patient immune responses rather than detecting the virus directly. The most common method used is ELISA, which is called the enzyme immunoassay. An IgM antibody usually appear in the first week of the illness and suggested that is a recent infection. An IgM response may be weak or active in the people who have a vaccine. Or IgG antibody indicate is a previous infection or vaccination. If we paired serum sample are available, a significant rise in an IgG level between an acute and a convalescent sample supporting the diagnosis of the recent infection. لو عايزين نستخدم ال IgG, we use 2 paired serum. One for the screening for the IgG. After that, next sample screening for the first is negative and the second is positive. That indicates this is a recent infection. IgG alone cannot differentiate between a vaccination and the past infection. But there is a cross-reactivity when we're using a serological test with other viruses like viruses, such as influenza virus, may occasionally produce a false positive IgM. Therefore, a serology test not always interpreted alone, usually interpreted together with a PCR result and the clinical finding or a clinical symptoms of the patient. How we can interpret the result of vaccinated patient? In vaccinated individual, IgM testing is much less reliable, not accurate. Why? Because 50 to 75% of vaccinated individuals with confirmed mumps may have a negative IgM result in some studies. How? This happens because the vaccination has already stimulated the immune memory, changing the antibody response during an infection from an IgM to IgG memory. In unvaccinated patient with acute mumps, IgM usually positive and the IgG rise over time. Opposite or in contrast, a vaccinated patient already have an IgG antibody before an infection, while an IgM remain negative. Therefore, the PCR becomes the most important diagnostic test in the interpreting of the result, not depending on the serological test, or we need to use all these techniques together to interpret the result. Genotyping and molecular epidemiology. Mumps has only serotype, but the scientists discovered that is 12 different genotype based on genetic difference which found in the SH gene. Genotyping is not used usually for the diagnosis in routine. It's used usually in the reference laboratory during an outbreak investigation. Used for to compare a viral sequence from different patients. Public health laboratories can determine whether by this sequence whether this cause belong to the same transmission chain or become from the different area. Genotyping does not always distinguish also differentiated between الوايلد تايب viruses اللي هي الجاية أصلا من الvaccines اللي هي جيلرين vaccine strain when illness develop soon after vaccination. Recently we found that the genotype of a gene G has caused most outbreak in the North America and Europe. حاليا لقوا إنه الجين genotype G cause an outbreak in the North America and Europe. Molecular epidemiology is therefore important for monitoring the virus circulation and supporting a public health surveillance. How we can prevent an infection of the mumps? The most effective way to prevent the mumps is through vaccination. The vaccine which used is an MMR vaccine. This is vaccine protect against the measles, mumps, or rubella. We can use the other vaccine MMRV, which is protect against the measles, mumps, or rubella, or varicella, or chickenpox. A mumps component which is found in the vaccine is a live attenuated vaccine, which is that meaning this is a weakened of the virus to stimulate immunity without causing a disease in the healthy individual. The vaccine usually stimulates both antibody-mediated immunity and the cell-mediated immunity, providing long-term protection. According to routine immunization, the first dose may be given at 12 to 15 months of the age, and the second dose given by 4 to 6 years of the age. During an outbreak, the public health authorities may be recommended a third dose of people at the high risk, such as the university student and the health care worker. This is a live vaccine. Because this is a live vaccine, we cannot give it to pregnant women or the patient or the people who with a severe immunosuppression. Vaccine effectiveness and outbreak. Okay. Although the MMR vaccine is highly effective, it does not provide complete protection. Why? Because there is waning of the immunity which is produced by this vaccine after a long time. One dose of this vaccine, it is have 78 effective to decrease an infection about the population. This means that a small number of vaccinated individuals can still become infected. This is the main reason for the recent outbreak اللي هي waning immunity اللي هو بيحصل شنو بيحصل decline في immunity after a long time. Here is examples of the cases of the patient. 20 years old college student presented with a 2-day of unilateral jaw swelling and low-grade fever. 3 dermatic contact reported similar sputum this week. 3 من أصحابه عنده نفس السيمبتومز. The student received 2 doses of MMR dose in childhood. The sample which used or the collected sample which collected from this patient اللي هي هناخد منه buccal swab urine and blood. Interpretation result depending on the result of the PCR, serology and symptoms. Reporting when this sample is positive, when the PCR positive or IgM is negative, that means what history of the patient have vaccinated. That means this patient is positive and reported directly to the national authorities who follow up supported provide the sample to the healthy laboratory for genotyping to link dermated cases and the formal outbreak control measurement. The keyword from the key message or summaries of this lecture. Mumps is one of the virus belong to family of Alparamaxviridae. It's involved negative sense RNA virus. mainly cause hepatitis infection, but can also affect the other part or other organ of body like the brain, testes, ovaries, or pancreas, or inner ear. Real-time PCR is the best method for diagnosing of acute infection, especially when the sample is collected during the first day after the symptom onset. IgM testing alone it is not reliable in the vaccinated individual. For that we need to use a PCR and integrate the PCR and serological test together. Genotype help the public health laboratory to investigate the outbreak and to follow virus transmission. Finally, to control the infection, how to prevent mumps infection, MMR vaccine remains the best method for the preventing mumps, even though immunity may decrease gradually over حتى والimmunity بتاعته بتنزل كل فترة تانية، لكن من الفاكسينات المهمة جدا جدا في control of the infection or control of the disease. Thank you for your attention. That is all for this lecture.